Namaskar, my dear students. Hope you are all fine. So, today we are going to discuss the next chapter of the unit genetics, and the name of the topic that I am going to discuss today is the mutations. You have heard of the term mutation as a change or something which is sudden. Today we shall be discussing the term mutation with respect to biological point of view. And my dear students, I must tell you that the term mutation for the first time it was introduced by a Dutch botanist Hugo de Vries in the year 1880 when he was studying the variations in plant Oenothera lamarckiana, commonly called as evening primrose. He found that in the subsequent generations of the plant some 834 plants out of the total population of 54,343 plants, they showed discontinuous variations, that is the variations apart from what was expected in the next generation, and he called these changes as the mutation. However, the term mutation as a discrete change for in the genotype was given in the year 1901 in his book Mutation Theory. And today, mutation can be defined as the sudden, stable, discontinuous, inheritable variation which appear in the organism due to permanent change in their genotype. What is meant by it? That a permanent change in the sequence of the nucleotide comes which altogether changes the genotype, and since this is a change in the genotype, it is inheritable and visible in the next generation, and this change is apart from the normal variation that appear in every generation. So this is sudden and stable and discontinuous and inheritable changes. Now, the product of the mutation is called as mutant. What is meant by it? That the plant or the animal which shows the mutation is called as the mutant. And what does the mutation bring in an organism? Mutation is a sudden change and it brings about the new traits in the organism. When new traits appear, they may be acceptable in the population or may not be acceptable. Whatsoever they are bound to bring variation in the organisms. That is, the organism shows a certain character which is quite drastically different from the parent population. And if it is acceptable and there exists a struggle for existence and survival of the fittest, and if the mutant is the fittest, it leads to speciation. What is speciation? Formation of the new species if that mutation is well adapted in the environment. And subsequently, because of above three factors, there is definitely the evolution comes into existence. Now I must mention here that mutation is not always acceptable. Mutation could be deleterious. It could be lethal, which means leading to the death of the organism. Of course, sometimes they are advantageous from the horticultural point of view, from the animal husbandry usages, etc. And at many times these mutations are quite neutral, that is showing no specific effect, and whenever the mutations come, they may be visible, they may be invisible. The mutation may be visible or invisible depends upon the fact that most of the mutations occur in the recessive genes and are not expressed, while some take part in the dominant genes and appear as the mutants. So, mutations are of various types but always bring about the change in the genotype. Now, depending upon up to which level the change is brought about, there are we have divided the mutations into various types. Broadly, mutations are of the two types, the gene mutations and the chromosomal mutations. Gene mutations are those which produce changes in the single gene, and chromosomal mutations are those which produce changes in the chromosome itself. A gene mutation normally alters the information conveyed by the gene. It alters the message. On the other hand, the chromosomal mutation only alters the number or the position of the existing gene, and it involves the modification in the morphology of the chromosome or change in the number of the chromosome. A still other type of the mutations which bring about the change in the number of the chromosome or the whole genome, they are called as the genomic mutations or the numeric mutations. I shall be discussing with you all these types one by one. To start with, let's talk about the gene mutations. Gene mutations is a change in the structure of the DNA which occurs at the locus on the chromosome, and because they occur only at the point of the nucleotide sequence, hence these are called as the gene mutations. Now, any change to one or the more of the nucleotide, because it brings about not the change in the structure of the chromosome but the sequence of the nucleotides which make up the DNA sequence, or any other rearrangement of the sequence will produce the wrong sequence of the amino acid and will make a protein which will be quite different. So, the gene mutations are the mutations at the level of the nucleotides. Now, gene mutations, they could be insertions, they could be deletions, they could be replacement. Insertion occurs when one or more nitrogenous bases are added. For example, here you can see in this sequence the parent genome is CTG-AGT, and in the next generation we see one additional nucleotide gets altered, which altogether changes the sequence of the amino acids and consequently the proteins which are to be synthesized by the subsequent nucleotides. On the other hand, deletions, they occur when one or the more nitrogenous bases are deleted. For example, this is the parent sequence, and in the next generation you can see this this A is not present, which shows one nucleotide is deleted. Insertions and deletions both change the subsequent code of the amino acids altogether, and whereas the replacement changes only one triplet and only one amino acids. So these mutations are also called as the point mutations or the gene mutations or the frame shift mutations, since by one insertion or one deletion whole frame of the nucleotides gets altered. Now next is the chromosomal mutations. Chromosomal mutations involve the chromosomal change in the number or the structure of the chromosome, because a part of the chromosome either gets deleted or duplicated or added. It can change, since there is a change in the structure of the chromosome, it can change the location of the chromosomes, location of the genes. It can alter the copies of genes, either it can reduce the copies of genes or it can add up the copies of gene depending upon the various conditions. There are four types of chromosomal mutations: deletions, duplications, inversions and translocations. I shall be discussing with you these various types of the chromosomal mutation with the help of this diagram. So here in this diagram you can see this is the parent genome, where we have divided the chromosome into segments. These A, B, C, these are not genes, these are the segments of the chromosomes. Now let us first discuss the deficiency or the deletion type of the change. Sometimes a segment of the chromosome breaks off and gets lost. For example, you can see the B segment is not seen in this next generation. Deficiency generally proves to be lethal or the semi-lethal. If the terminal segment of the chromosome is lost, it is called deficiency, and if the intercalary segment is lost, it is called deletion. Both the segments of chromosomes which are cut after losing the part, they can rejoin and deleting the intercalary segment. Now, similarly, now we will be talking about the duplications. Here is the parent chromosome, and here you can see the B has got duplicated. Actually, B which was reduced from here, cut from here, has got inserted over here. Now what happens in duplications? The deleted chromosome segment is attached to the normal homologous chromosome. Here a gene or the many genes are repeated twice or more number of times in the same chromosomes. They are more significant than deficiency. Next is the inversions. Sometimes as a result of doubly broken, this inversion means getting rotated, the genes are equally present, does not alter the size of the chromosome, but their orientation is altered. The center piece is inverted. For example, a chromosome with the genes A, B, C, D, E, F is broken from B to C and from D to F, and the portion is turned through 180-degree, resulting in the gene sequence. Now instead of A B C D E F, it reads A E D C B F, because a portion from E B C D E that has got cut and inverted at 180 degrees. This is called as inversion. The last one is the translocation. Now what is translocation? If the broken end of the chromosomes join to non-homologous chromosome, it is called as translocation. An exchange of part between two non-homologous chromosomes is called as reciprocal translocation. As a result, one chromosome represents deletion while the other shows the additional component. So these are the various types of the chromosomal mutation. You can even better understand from this diagram. This is the original chromosome. This is the result of deletion of B. This is the result of duplication of B. This is the result of translocation, and this is the result of inversion. So these are the chromosomal mutations. Now the next that we shall be discussing is the ploidy, or the genomic mutations, or the numerical changes in the chromosomes. Numerical changes in the chromosomes are the variations in the chromosome number may be due to aneuploidy and euploidy. There are the two types of the genomic or the numeric changes, aneuploidy or the euploidy. Aneuploids represent the number different than a multiple of basic set of the chromosome. That is, if we generally consider 2n is the basic set, then the altered would be 2n plus 1, 2n plus 2, or 2n minus 1 and 2n minus 2. If the chromosome number is lesser, it is called as a hypopolyploidy. If the number is more, it is called as hyperpolyploidy. Depending upon how many chromosomes, it could be trisomic, tetrasomic, or monosomic and nullisomic. Similarly, in case of euploids, the somatic chromosome number in the euploid is the exact multiple of the basic haploid number if the organism has an additional complete set of the genomes, and when the genomes get repeated, this is called as the euploidy. If the genome of the same species gets repeated, it is called as autopolyploidy. If from different organism, it is called as the allopolyploidy. We shall be discussing these types one by one and the basic differences between them. Let us first discuss the aneuploids. If any chromosome number that is not euploid, u means true, ploid means multiple, that is if the resultant chromosome number is not the exact multiple of the basic set, it is called as the aneuploid. It is the abnormal number of the chromosome with either one extra or the two extra or one lesser or the two lesser. Now, in case of aneuploids, we have several conditions. Aneuploid can be either due to loss of the chromosomes or due to the addition of the chromosomes. The Due to the addition of one or the more chromosomes to the complete chromosome complement, we get hyperploidy. The condition is monosomic if it is 2n minus 1. It is nullisomic if the whole chromosome pair is lost, that is 2n minus 2. And the examples of hypoploidy are the this hyperploidy includes trisomy 2n plus 1 and tetrasomy 2n plus 2. Trisomics may originate spontaneously due to production of n plus 1 type of the gametes due to rare non-disjunction of any of the bivalents. Now the next we shall be discussing the euploidy. The somatic chromosome number in the euploids is exact multiple of the basic haploid number in the euploids of an organism, and the organism acquires the additional set of the chromosomes over and above the diploid component. Now these there according to if their single set is called as the monoploid or the haploid, whereas the two sets which is the normal course it is called as the diploid. If there are the three sets it is called as triploid. If there are four sets it is called as tetraploid, pentaploid, six sets hexaploid, and if there are the organism with seven sets is called as the heptaploid. Now several uh, ploidy levels can be obtained, but whatsoever is the polyploidy, it could be autopolyploidy where the multiplication of one basic set of the chromosomes occur, and it could be allopolyploidy if the combination polyploid is not the result of one species but two dissimilar chromosomal sets. When they are added, it is called as the allopolyploid. Now let us find out the differences between the autopolyploids and the allopolyploids. They can be summarized as follows, as you can see in this table. Autopolyploids refers to the type of the polyploidy where the chromosomal complement consists of more than two copies of the homologous chromosome, whereas the allopolyploids, the polyploidy where the complement chromosome set are of the two copies of the chromosomes are derived from two different species. The autopolyploids arise by the fusion of the gametes of the same species. Allopolyploid arises from the fusion of gametes of the different species. Autopolyploid cells contain homologous chromosomal sets. All the chromosome sets of an alloploid polyploid are not homologs. Autopolyploidy generally arises due to the non-disjunction of the chromosomes, whereas allopolyploids are generally due to hybridization experiments. Hence, meiosis is not perfect part of the cell cycle in the autopolyploids, whereas the allopolyploids may undergo meiosis. Autopolyploids are very commonly exploited in the crops of the wheat, oat, sugarcane, potato, banana, etc., whereas the allopolyploids are seen in the wheat and also in some animal species. The example is the mules. Dear students, the ploidy level or the chromosomal mutations are the frame shift mutations. If they are of some use to the mankind, then they can be artificially induced, because it is found that the Spontaneous mutations are the mutations which occur in nature. Their rate is very slow and at all loci in all the organisms. Muller's experiment decided that the mutation rate is much higher in the progeny of the Drosophila, which was treated with the X-rays, and hence the concept of artificially raising the mutations was introduced, and with the help of the artificial agents called as the mutagens, if it is of any use. Mutagen is a physical or the chemical agent that causes mutation, that is changes in the genetic material, usually DNA of the organism. Now the substance which causes mutation is called as the mutagen. However, all the mutations, they are not advantageous. Mutagens may cause cancer and are likely to be known as the carcinogens then. Now, whenever the mutagen is applied, it could produce various mutants, and out of which we could choose the mutant of our choice. Various mutagens are used, and broadly we have classified the mutagens into physical, chemical and biological mutagens. First of all, physical mutagens, which which could be either some sort of radiations or some temperature treatment. First, let us talk about the radiations. All types of the energy that can change the chemical structure of the gene are the chromosome induced mutations, for example X-rays, alpha rays, gamma rays, beta rays, cosmic rays, ionizing radiations, whichever can alter the structure of the DNA, thereby disturbing its replication and DNA duplex, is called as the radiation physical mutagen. Next is the uh, heat or the temperature. A rise in the temperature may lead to the disturbance in the gene that cause and thereby causing the mutation. Rarely low temperature treatment may also cause mutations, and it is seen in the paddy. The low temperature treatment causes the mutation. Then let's talk about the chemical mutagens, which could be base analogs, some intercalating agents, alkylating agents, deaminating agents and metals, etc. What are the base analogues? The base analogues, the two commonly known base analogues are 5-bromouracil and 5-fluorouracil. Actually, analogues are the substance which can replace themselves for a particular nucleotide. And these two analogues, that is 5-bromouracil and 5-fluorouracil, they can replace thiamine, and pairing of the adenine they get thus gets altered and they pair with the G. It disturbs the replication, transcription and translation mechanism of the DNA molecule. Then next is the intercalating agents, and some of the chemicals like CH2Cl2 or nitrogen mustards causes addition of the methyl group to the nitrogenous base, thereby disturbing the normal course of replication of DNA and transcription, and thus bringing about the mutation. Then are the dyes. Certain dyes may play a role in the bringing about frame shift mutations. Such dyes are the acridine orange and the proflavin, which can play a role in causing the mutation. That is, they can frame shift mutation can occur because such mutation form the nonsense polypeptide chain or the codon. Deamination is another method of the bringing about the mutation. Some chemicals like nitrous oxide deaminate. Deaminate means removing the NH2 group or the amino group, the nitrogen bases, and therefore change the codon of the DNA. Therefore, they bring about, as a result of deamination, replication, transcription, translation are disturbed and the mutation occurs. Then we shall talk about the biological agents which consist of some transposons, virus, bacteria, etc., which integrate with the normal genome of the organism and alter the normal genome and thereby bring about the mutations. So these mutagens are being exploited to various extents to produce the varieties of our own choice or obliterating some undesirable characters from the from the population. So that's all for the mutations. Hope you have well understood this topic. If you have any query, you can ask me anytime. Otherwise, thank you and have a nice day.